Today, I will be discussing the protein KRAS with the mutated glycine AA number 12 that is replaced by cysteine. The KRAS gene provides instructions for making a protein called KRAS that is part of a signaling pathway known as the RAS/MAPK pathway. It is called KRAS because it was first identified as causing cancer in Kirsten rat sarcoma virus. The KRAS gene belongs to a class of genes known as oncogenes. When mutated, oncogenes have the potential to cause normal cells to become cancerous. The KRAS protein is a GT-PACE categorized as an enzyme, which means it converts a molecule called guanosine triphosphate, or GTP, into another molecule called guanosine diphosphate, or GDP. Inactivation of the active KRAS occurs upon GTP hydrolysis to GDP. KRAS activates the GTP plus H2O, which is water, yields KRAS inactive GDP and an inorganic phosphate. Its signaling activity depends on its level of enrichment on the plasma membrane. This is why it is a plasma membrane anchored protein. To transmit signals, it must be turned on by binding to a molecule of GTP. This protein is turned off when it converts the GTP to GDP. Healthy KRAS basically tells the cells when to divide. They can also help cells to mature and take on specialized functions. RAS proteins are binary switches cycling between on and off states during signal transduction. KRAS switch regions known as switch 1 and switch 2. These switches from the binding interface for effector proteins as well as for RAS regulators. Hyperactivation of RAS signaling, which may occur via a direct mutation of RAS or indirectly via other proteins in RAS pathways, plays a significant role in cancer and in particular rare diseases. KRAS mutation in the G12 hotspot will be the main one I will focus on. This G12C is a single point mutation with the glycine to cysteine substitution at codon 12 in the KRAS gene. This substitution favors the activated state of KRAS. Hence, when KRAS is stuck in the on position, it allows cells to grow uncontrollably and activate the downstream pathways. This is cell autonomous signaling, which is when a product is involved in signal transduction, the process by which a chemical or physical signal is transmitted through a cell as a series of molecular events, which is common in proteins. When a protein is bound to GDP, it does not relay signals to the cell's nucleus. This results in the excess production of KRAS proteins, which causes abnormal or too much cell growth. As aforementioned, this rapid production of cells can cause some of the cells to become cancerous. The G domain of KRAS forms the basis of biological functionality of the GT-PACE proteins. This domain includes 6 beta strands forming the protein core, surrounded by 5 alpha helices. With the G domain, KRAS has a flexible C-terminal structural element named the hypervariable region, or HVR, which plays a crucial role in anchoring RAS to the membrane. KRAS mutation hotspots in cancer are located in a phosphate binding loop, also known as P-loop, or in switch 2. My model does not effectively show the P-loop or switch 1 being activated. It also does not include MG, and it does not show GTP, which is the guanosine triphosphate. The natural unchanged form of the gene is called wild-type KRAS. Mutated or changed forms of the KRAS gene have been found in some types of cancer, including non-small cell lung cancer and pancreatic cancer. The main type of cancer that I would be focusing on is lung cancer. This KRAS mutation tends to occur more in patients of Western European descent and current or former smokers, but it has been found in people of many different backgrounds. Around 20 to 25% of lung cancer patients have some kind of KRAS mutation. Three of the commonly used methods for KRAS mutation detection in clinical samples include nucleic acid sequencing, real-time PCR with melt curve analysis, and allele-specific PCR with various modes used to distinguish mutant from wild-type sequences. There are a couple of treatments for people with the KRAS mutation. Surgery, radiation, chemotherapy, immunotherapy, or a combination of all the above can be an option for those with KRAS positive lung cancer. Patients with the KRAS G12C can take the targeted therapy pill called sotorasib if their cancer stops responding to chemotherapy or immunotherapy.